Okay, so now that you watched that video about solute potential and water potential, we can walk through this equation. And in that video, they used the term solute potential. Here, we're going to use osmotic potential, but effectively think of them as the same thing. So this equation, when you look at it, just, I don't know, makes my eyes turn sideways. This is the Greek symbol PSI. He pronounced it psi. I believe psi is the correct way of pronouncing it, but however you want to is fine. And this is the uppercase psi symbol. So what we're saying is that water potential is the sum of osmotic potential, pressure potential, and matrix potential. So osmotic potential we just looked at. Again, that's what they called solute potential. Water moves to the more negative value. So if we calculate the water potential of a root compared to the water potential of the soil, we would hope that water potential of the root is lower, more negative, than that of the soil. So water moves from the soil to the root. So water follows the lowest number. It follows the lowest potential. Pressure potential, as described in the video, is atmospheric pressures. Frequently we think about in things like tall trees. Now we've got a pressure potential the tree has to overcome, right? If you're 100 feet tall, it takes a lot of pressure to get the water up there. And matrix potential we talked about is typically associated with either the stickiness of the cellular structure to hold on to water, or frequently in soils, typically more humus, more organic content, finer grain, has a higher matrix potential, which is a negative value. So you could have roots living in sand and roots living in a dark organic matrix, and that dark organic matrix is going to have a potentially higher matrix potential, which here is a negative value. So it's going to be harder to move water from that soil that has a high matrix potential compared to soil that has and by high I mean a more negative matrix potential, compared to something like sand, which has a matrix potential closer to zero. And so water always flows from a higher water potential to a lower water potential. When we look at some examples, water potential is typically a negative. So we're flowing, when we say from higher to lower, what we really mean is flowing to a lower number, which would be a more negative number. And again, because osmotic potential and matrix potential are negative values, we really wind up with a sum that's in the negative. We'll walk through that in the next slide, and hopefully this makes more sense. But it's a way of thinking about how plants absorb water. So here's a graph that kind of walks through this here. So if we start with the soil, this has the highest water potential. This is MPa, which stands for megapascal. It's just a pressure unit. And I believe 1 megapascal equals about 10 bar. And so we're looking for water flowing from the highest water potential to the lowest water potential. So in this system, our highest water potential is the soil. Roots have a lower water potential, in this case a more negative water potential. So water would move from the soil to the roots. And then the xylem have a even lower water potential, so water would move from soil, root into the xylem. and then out to the leaves. Now there's a huge gradient between the leaf value of negative 1.8 megapascals and the atmosphere, which in this case is negative 100 megapascals. So the gradation between the leaf and the atmosphere is quite large, which is why plants have to have a number of adaptations so they don't lose all of their water to the atmosphere. And there's another couple examples of this that we'll walk through, but this is one way that water potential is put into practice. And so this can help us understand why water moves and what soil conditions are conducive to water absorption by plants. And the atmospheric water potential is related to relative humidity. And if it's less than 98%, the water potential is low relative to organisms. So unless humidity is 99 or 100%, there's typically a strong gradient between the atmosphere and relative to organisms. And terrestrial organisms have to have barriers to resistance to water loss. These barriers include skin or waxy cuticles of insects or plants. And so, as well as the stomata, just the nature of terrestrial organisms, there has to be some barrier, otherwise we would just lose massive amounts of fluid. You can imagine if our skin was more like a pure sponge, we would lose a lot of water every day. Here's a diagram talking about the water content of soil, and it's a few things. Obviously precipitation, and that's the amount of precipitation that makes its way into the soil is called infiltration. Infiltration rates vary, typically associated with development. So if you have more development, you have more impervious surfaces, concrete rooftops, roads, etc. That reduces water infiltration, reduces the amount of water that makes its way into the surface sediments, and that creates more overland flow, as well as bad forestry practices, denuding the environment, removing plants that slow water down and help it infiltrate into the shallow sediments, ultimately resulting in percolation and development of a deep groundwater. Now, groundwater, on the one hand, I used to think of groundwater as deep. You hear stories in California where they're drilling down hundreds and hundreds of feet to tap into deep groundwater so they can irrigate almonds. In Whatcom County, we have what's called the Blaine-Sumas Aquifer. And if you drive through Whatcom County, you actually see a fair number of ponds and wetlands. And that's mostly driven by our groundwater. So throughout most of the Blaine-Sumas Aquifer, which is basically North Bellingham to the border, and as far east as a bit east of Sumas, that aquifer is frequently 10 feet or less in depth, meaning it's groundwater is right there. And so this relationship between precipitation and groundwater is pretty tightly linked in that example. We have a fair number of deeper groundwater pockets that are down in the order of thousands or hundreds of feet as well, but we have the surface layer that's very, very shallow. And so as water infiltrates, it's taken up by plants. There's some evaporation loss from the surface, and then transpiration, that loss of water from the plants back to the atmosphere. And some water winds up in what we call surface water, rivers, etc., ponds, lakes. So plants have rigid cell walls of cellulose that allow the development for turgor pressure. As water moves in the cell, it expands and presses against the cell wall. Turgor pressure helps give form to plants, and it's important for us for growth and promoting cell division. Wilting or loss of turgor is a sign of water stress, and so we'll look at that. This kind of goes back to the 100 series of biology, but plants require this stiffness. They have a cell wall that provides some structure, but they need water to come in to sort of support that structure. And we're all familiar with a wilting plant that is low on water. It can't support itself anymore. This is the mechanism for that the plant cell typically has more solutes, and so compared to the environment, so water wants to move into that space, and that helps exert external pressure on the cell. So it has the cell wall, but it needs water pressure to push out against that cell wall to give it rigidity. Here's some examples of what cells can look like under different conditions. And so the first one is hypertonic, and so here there's more solutes outside of the cell than there is within the cell. That causes water to leave. If you take a freshwater plant and put it in salt water, the water will leave. Isotonic's an ideal situation where, actually it's not. So isotonic would be there's an equal number of solutes in the cell and in the environment, which would allow an even exchange of water. So there's still water within the cell, but not enough to exert a positive force outward. So plants really want to be in a hypotonic system where the water has fewer solutes compared to what's inside of the cell, and that will help keep the cell turgid. Turgid, giving it turgor. It's like turgor. It's like grr. So you want more solutes in the cell compared to outside. So it's important to have, basically fresh water. Ideally not salt water. So plants lose water via transpiration when the stomates open to allow CO2 to come in. That's one thing we didn't get into, and that's more of a biology, like the C4 versus C3 plants. It's not something we're talking about now, but plants need carbon dioxide to, undergo primary productivity. And so the stomata is one way, or the primary way that carbon dioxide comes in. So they have to open the stomates to let carbon dioxide in. When that happens, water vapor goes out. As the leaf loses water, the water potential in the cells decrease relative to the stem. In xylem, so water moves from the stem to the leaf. Water moves from the soil, which has the highest water potential, along the water gradient. So going back to the diagram up here, highest water potential, lowest water potential in the system, in the terrestrial system, and then the lowest water potential is the atmosphere. And so this gradient is what's driving water up. Root uptake lags behind transpiration rates during the day, so the plant water content decreases. You can see this sometimes in, check an example, like maybe tomato plants, where they can wilt a bit during the day if there's not enough water. At night, the stomates close and the plant water increases until it reaches equilibrium with the soil water potential. The lack of rain decreases soil water. Plants can lose turgor pressure and dehydrate. So here's another image showing relatively the same thing. This is not showing the water potential of the air. But during the day when the stomate's open, transpiration results in a gradient of water potential from the leaf to the stem to the roots. And so what's happening is water is leaving the leaf. The solutes within the cell are becoming more concentrated, which is further driving that water potential and pushing water up the plant into the leaf and creating this larger water gradient. At night, when the stomates close, there's not a lot of loss of water via transpiration, and so eventually the cells fill back up and come into equilibrium. And so ideally, here we've got root, stem, and leaf all at the same water potential value, in this case 0.5 megapascals. This is a one way of thinking about it. And it's kind of interesting. So we'll look at transpiration rates first. And so here's a graph of transpiration rates during the day. Transpiration rates are higher. At night, they're lower. Kind of wish these bars went all the way up. So during the day, when you have a lot of transpiration, looking at, I don't like these colors, but looking at the leaf, the water potential goes way down a bit on the root, and the soil potential stays the same. Now, what this is looking at is soil depletion, or depletion of soil water. And so this is, imagine you have a tomato plant in a 5-gallon bucket and you put it out on your back porch and you don't water it for a week. This is effectively what it looks like, where water's being used and the water potential of the soil is getting larger and larger. As there's less water in the soil, the water potential gets higher. And plants are able to still deal with that and pull some water out by increasing their water potential as time goes on to a certain point. But eventually they won't be able to overcome that. There's not enough water in the soil to be easily extracted. So you can kind of see this variation from day to night, and the root values in particular kind of sagging down. Yeah, so eventually this is going to death. This is kind of an interesting way of thinking about this. This is the allocation of growth to roots versus shoots and associated with precipitation levels. Root to shoot ratio. Sounds kind of funny, but what it basically means is the ratio of below ground biomass compared to above ground biomass. If you have a lot of below ground biomass compared to your above ground biomass, you're going to have a high root to shoot ratio. Trying to give a terrestrial example of that. I'm trying to think of a good one. Eelgrass makes sense to me, but that's not a very good terrestrial example. I'm weak on my terrestrial plants. But something with a large root mass and a small vegetative piece up top. Grasses, perhaps. Think about things that you can cut the entire above ground vegetation off and it sprouts right back up. These are typically associated with areas that have lower amounts of precipitation. So if we look at precipitation in millimeters along the X and then the root to shoot ratio along the Y, this grouping up here, they have a much higher amount, about 25 times more below ground biomass compared to their above ground biomass in areas with very little amounts of rain. So the mechanism here, or one rationale of explaining that difference, is that in areas with low amounts of rain, plants need to have a massive root bed to do a few things. One, have a big root bed to absorb as much water as possible. And then you can also imagine perhaps the vegetative part, the shoot part of the plant, will die off maybe in the summer when it gets too hot. And then there's enough energy stored in the below ground root mass where they can re-sprout that again. So it's kind of two of those things, but typically the idea is having more biomass in the roots in dry soils helps provide water uptake capacity to support that above ground biomass, which is your photosynthetic biomass. This is kind of an interesting thing to think about, which is the same organism can have to deal with water loss and solutes differently based on life stage. So in the example of a organism that has an aquatic life stage and then a terrestrial life stage, a lot of dragonflies, for example, but a lot of our aquatic insects are actually the larval stage of typically a winged insect. So in the aquatic larval stage, the organism has to deal with losses, covering the losses of water across the outer covering, diffusion or secretion across the gills, excretion or defecation. It's able to gain water by food and drink. It's able to uptake water across its outer covering, and it can gain water through oxidative metabolism and uptake water across the gills. Now, when they become an adult in their terrestrial adult stage, now they're losing water through net respiratory evaporation. So just the same as when we breathe in and out, there's a small amount of water loss. The same is true across the gills or breathing structures of insects. Net movement of water across the outer covering. So insects have, some sort of covering that typically protects them from water loss. Gamete or egg production, all of that is a net loss of water, as well as excretion and defecation. Here, they have to take in food as well as potentially drink water. Still some gain through metabolism, and then net water vapor absorption if possible. So it's just kind of interesting to think about the differences between life stages and how organisms deal with water loss and gain. We're going to run through a couple types of organisms, going back to our prior terms of isoosmotic, hypoosmotic, and hyperosmotic. So marine animals tend to be isoosmotic to the seawater, meaning that their concentration of salts and solutes is about the same within the organism versus the environment. Some invertebrates exchange solutes with the environment to maintain osmotic balance, so they can bring in or excrete solutes to maintain that balance. And so they have to selectively control and exchange specific solutes because some are toxic at concentrations found in seawater. Things like sulfur concentrations that can be toxic if they build up. So they can't just take in any solutes, they have to be selective in those. Cartilaginous fishes, including sharks, rays, have a blood solute concentration similar to seawater, where the bony fish, teleos, evolved in freshwater and later moved to the oceans. So their blood is hypoosmotic to seawater. So here they're being clear in how they're explaining it. So here they're saying hypoosmotic compared to seawater. Less solutes compared to seawater. Hypoosmotic to seawater. So they tend to lose water and gain salts. So water tends to move out of their body into the seawater, which has a greater amount of solutes. And then salts tend to move in. So they have to be able to deal with that. And so here's examples of two different fish living in different environments and how they're able to deal with that. The first is what looks like a cod, where it's drinking water. It has osmotic loss through its gills and then urine, and it's gaining solutes from the seawater, and it has to secrete through the gills calcium and chloride, and then magnesium and sulfur through through its urine. Where for freshwater fish, because they're typically isoosmotic, they're not dealing with having to excrete solutes. They're more or less in osmotic balance. So they're gaining some water from their gills, they're excreting some from the urine, they're bringing in solutes from food, they're uptaking it through their gills, diffusing it through their gills, and expelling it through urine. Marine mammals, whales, porpoises, etc., minimize salt uptake by not drinking seawater, and they produce urine that is hyperosmotic to seawater, a greater concentration of solutes, so they produce really concentrated urine. Freshwater animals tend to be hyperosmotic to the water. They tend to gain water and lose salts. Solutes are taken up in food across the gills and across against the osmotic gradient. Excess water is excreted via dilute urine. And terrestrial animals must exchange gases in a dry environment. Evaporative loss is minimized by skin or by living in moist environments. Think about worms. They dry out when they're on the concrete, but they're fine when they're in the soil with a fair amount of moisture. And resistance to water loss can limit gas exchange. So that's it for this one, and this will wrap up chapter 4 for the lecture portion.